And for the next speaker, we're going to move back to the Netherlands, the Dutch peat soils. So please take the floor.
Nice to be here. My name is Tolunay. I'm a PhD candidate at Utrecht University, and I am working one of the land subsidence mechanisms in Dutch peatlands that you heard of it, and at the project called Living on Soft Soil Loss. Where's my pointer? Ooh, that's something else. During the conference, I have heard a lot of land subsidence mechanisms, and mainly focusing on physical mechanisms. But today, for a change in this presentation, I will take you another world, a living world beneath our feet, the soil microbial community living in the peat soils. And yeah, we heard about peat oxidation, but I'm not going to use the term peat oxidation for this presentation. I'm going to use microbial-driven decomposition or decomposition of organic matter. The reason is that I think there is some misunderstanding of what peat oxidation or what oxidation means. Because of a chemist who named this chemical process oxidation, the first thing we think, there must be oxygen. That is true if we are talking about an oxic peat zone. Then the microbes living there adapted to a condition where oxygen is required to oxidize the peat. But there are some microbes, smart and quite surviving the harsh conditions, can live in the anoxic peat zone, then they don't need oxygen. They can use alternative sources such as iron, sulfur, manganese, whatever you call it. But still, peat can be oxidized under water-saturated conditions. Therefore, I'm putting the peat oxidation aside and more using the decomposition of organic matter. And why do you oxidize the peat? The answer is very simple. They want to survive like other living organisms. And to be able to survive, you need food. And the food is organic matter, basically. And in the peatlands, as an organic matter source, we have plant materials. And these plant materials, of course, come from different plant species, which means that they have different chemical composition, different organic matter structure, could be size and shape can be different. And then these microbes need to think how we can eat the peat. So we need to figure out a system where they can take these big molecules inside their body. And they come up with a very smart solution, not all of them, but some of them. And they made these exoenzymes, extracellular enzymes, and they produce these enzymes inside their body, and they just excrete those enzymes into the peat matrix. And what those enzymes does, take the big organic molecules and make them smaller, chop them, and make it available for the microbes to grow, survive, reproduce, and then as an end product, of course, they produce carbon dioxide, nitrous oxide, methane, and we have the peat loss and the land subsidence. And just for, I got a question from Ramon, the peat oxidation is chemical. So when you look at this here, the peat oxidation or oxidation is a chemical reaction catalyzed by an enzyme which is produced by microbes. So it is biochemical reaction, just for you. So I'm now gonna give you not hardcore biology course, but just to let you to know what kind of extracellular enzymes that we have. So we have two hydrolytic exoenzymes and oxidative exoenzymes, and they are, like the hydrolytic ones are required water to function, and the oxidative ones required oxygen to function in theory. That is important because all the steps that determines the decomposition rate is depending on these drivers that we see in each steps. Could be oxygen, microbial community, or the botanical origin of the organic matter. And I'm just going to focus on two things, the oxygen and the botanical type. Why? Because we're in the Netherlands. So the oxygen is a very important driver because we are managing the water in Dutch peatlands. And what it creates 3 layers in the peat soil. The top layer, we have an oxic zone where the oxic microbes can survive. And we have this deep zone, the water saturated zone, very little or no oxygen. Then we have totally a different microbial community. And we have this fluctuating zone, which the oxygen can be available sometimes. This is very important for the microbial community. What kind of, who is there, who is living there, changing in these 3 zones. And that determines how much carbon dioxide is going to be released to the atmosphere. And of course, the peat loss, land subsidence. And the second driver that we pick is the peat material. So we divided the peatlands into 4 based on the major plant material in the peat matrix. We've selected reed, wood, sedge, and sphagnum because they have different chemical composition. Some of the organic matter are easier to degrade by those enzymes, microbes. Some are more difficult. You need more time, more energy. And we wanted to know the decomposition rate diverse between different sites that we're going to check. Which brings us to research question. So what we want to know, we want the role of the peat material, these 4 different plant material, whether it determines or how it determines the rate of the oxic, but also anoxic decomposition rates from the Dutch peatlands. And for that, we visited 4 field sites in the Netherlands. So Zegveld, some of you already visited during the, I think, excursion day, that has a wood peat in its matrix. And we have Ralveen. This is the pointer. Yeah, this is Zegveld. And this is Ralveen. And we have Banskotten in the middle, a sphagnum pit, and the Onlanden on the top that has reed peat in its matrix. And we collected our samples deeper, more than 120, so we know that they're water saturated, they are well preserved, and we always treated them anoxically, so we brought them from the field to the laboratory conditions under anoxic situation. This is me, how I work. So since oxygen is the main driver, it's very, very important that we know that we are working under anoxic conditions. So we got this anoxic chamber. It's not easy to work with, but yeah, it gives you a back egg, neck egg, every egg you can think of. But still, it also gives you lots of information, what you want to know. And I'm gonna talk about the results, but first I'm going to tell you what we did with these collected samples. So we split them into 2, and we incubated them under oxic or the anoxic conditions. And we know that the time is a very important element for the decomposition of organic matter. So what we did after 24 hours, we call that T0. We measured our parameters, oxic and anoxically, and then after 15 weeks, we measured them again. And what we measured is, first of all, basal respiration rate, so how microbes are active, how much carbon dioxide they are producing, and the six different exoenzymes, which has different functions in the system, and then some chemical characteristic of the peat that we wanted to know. Results. So, okay, carbon dioxide emissions, how much carbon dioxide is produced. So in this graph, looks like complicated, I'm gonna make it very simple. So each block you see are the different field sites that we collected samples, so sphagnum peat, this is the reed peat, the sedge, and the wood peat. And then you will see the gray ones. Those are the ones that the first time we measured, so 24 hours incubations. And the yellow ones after 15 weeks that we measured. And if you look at here, AN indicates the anoxic conditions that we incubated our samples, and the oxic ones are the OX ones are the oxic samples that we incubated oxically. So the main conclusion of this graph is that, first of all, each site has a different response to oxygen exposure in different time steps. This was the raw vein, is the sedge peat, has an interesting response. When we first time expose all of them on the oxygen in 24 hours, the sedge peat responded very sudden. So the microbes just went crazy and become super active, and the emission rates increase drastically. And then something happened after 15 weeks. The most stable, the most conserved community was the sedge peat. So the sedge peat stay more or less stable, whereas the other ones this time went crazy. So this shows the vulnerability of peat in terms of the microbial activity is very different wherever you go, which ecosystem you are looking at it. The other thing that is mainly overlooked, or underlooked, is the anoxic decomposition rate. So the production of carbon dioxide also occurs anoxically. It is there, it is less lower than the oxic one, that's true, but it is something that in long term can contribute to land subsidence rates that we need to check more into detail. What are the other elements, environmental factors, triggers these anoxic community to get more active? Could be the water chemistry one of them. I told you that I also check some enzymes, and many enzymes we check to see whether they are correlating each other. Luckily, they are. So there is a positive relationship between these enzymes that actually help us to, if you want to monitor a site, and if we decide several parameters to monitor, we don't have to spend so much time, money, investment on 5, 6, several enzymes, but we can pick one representative enzyme that we can use it in the monitoring programs. And the other interesting things, well, more or less expected, the pH is a strong driver of the respiration rate of microbes, but this usually you would expect a pH increase and the microbes get more active. We see a very strong negative effect, but it's because of one site, the reed site, that has a pH drop from 5 to 2, which actually indicates that that was not a microbial activity, but some chemical-driven reaction that leads to carbon dioxide emissions in the system that we are looking into in detail now. Take-home messages. So first of all, the decomposition rates vary between peatlands based on the dominant plant material, but it's so clear that we only collected data from one site peat, one site woods. We need more data, so now we have the database to add more and to tell something about the dominant plant material make different, and maybe we should consider that while we are approaching the land subsidence rate as well. The other important take-home message is that anaerobic microbial community is there, they are active, and their activity can be triggered with the human activities, such as the water chemistry. Some of the Dutch peatlands has iron and sulfate in its body, and that can trigger the microbial community because they can use these external sources to get more active, to make babies and then make more carbon dioxide or methane and nitrous oxide. So we need to look into that too. And we are actually already doing it. So the first thing, we already completed the understanding the range of decomposition rates in Dutch peatlands. So now we have more or less the understanding of it. And we are on the halfway to understand a bit more drivers of this decomposition rate. What we did on the microbial community and substrate focus, we just got a 1 meter of peat core, where the top part is oxic and the lower part is anoxic. We just flip them and put back in the field. And after 2 years, we're gonna check what's going on there, because we want to see how microbes are shifting and affecting the land subsidence rates and the greenhouse gas emissions. And the other one is the pore water chemistry. So our focus is iron and sulfate. We want to know if we provide these sources, whether they get more active, and we have some proof that they do, and that will come in the future, maybe the next TISOLS. And the last part, which is something that also not very much studied, that looking at the interactions of lens subsidence mechanisms. So we want to understand whether the physical properties, physical changes affecting the biological or the other way around. And I am hoping to do that with my colleague Pepijn van Elderen, also sitting in this room now. And we're gonna look into whether the interaction really important if we're gonna check the lens subsidence rates in the future. And I would like to thank you all that you were here and you listened my story, and I would like to thank you all my colleagues that they contributed somehow in this presentation and in this work. Thank you.
Thanks so much for giving a face, literally, to oxidation. I think that's a nice perspective for the people in this room. Questions? For the sake of walking, I'll start with you, Roman.
Yeah, thank you very much for your presentation. Do you now, should I see this as a proposition that we should not use the word oxidation anymore? Is it still valuable, or how are we going to communicate about this in the future?
Yeah, I'm fine using the peat oxidation, because oxidation is a chemical reaction that is named by a chemist, and it is there, and we just need to know what does it mean. Mm-hmm. So we shouldn't say that peat oxidation equals the oxygen intrusion, and the oxygen creates peat oxidation, but oxidation is just the decay of an organic matter. It's a chemical reaction. So we just acknowledge that oxygen is not required to have a peat oxidation. So I think that knowledge is, if just we spread that news, we can use the peat oxidation.
Yeah,
no
well, that's good news. We can,
you can relax.
Understand each other. Yeah. The second question that I had is so your metric that you're producing is respiration, and how can I make a direct translation between respiration and subsidence or volume change?
Yeah, actually, that is something that we are trying to do, and I think Gilles already showed something in his presentation that they did more using the organic matter content as well. And I'm hoping maybe with the data I have, I can try to reach something, but the amount of data I have is little. So my first step, I think, a bit expand the data that I have, and then convert it into a lens subsidence rates.
Thank you.
Thank you. Very nice work. It's really very good. I have one question. I suppose that in some way you start speaking also about temperature. I'm not an expert about microbes. I don't know anything, but what I know is that let's call it oxidation. Also today, oxidation is really strongly dependent on temperature. So did you take into account this factor when you also make your lab test? Did you also control the temperature? How you work with these factors?
That's a good point. The reason I did not really mention, because that's the factor that we stabilize. So since we were doing a lab work, we can control the temperature and moisture content. So we kept them stable, and 23.5 degrees that we kept them. So all results based on one temperature. But that's true, microbes are very sensitive to temperature changes. Now we have a field samples that we are doing a field experiment. Most likely it's changing, but we can only take one snapshot the time that we are measuring, and to make it compress, to be able to check them, we need to just keep them at the same temperature while we are measuring the parameters in the lab again. So we always fix the temperature to be able to compare the results.
Do you think that this normal rule that we use, so we have the exponent, so the temperature goes to the exponent of this microbial activation, is something that it is, let's say, that works, or we have in some way to remove and to move to something more new or more scientifically based on your expertise?
Yeah, the temperature should be there. There is no way to remove it. It's very important for the microbial community. But we need to start from somewhere, first understanding how microbes behave. It's more complicating than we thought. So now we are trying to keep everything as stable and at the same level as possible. So remove or change one driver that we want to check, and see it. And I think, I don't know, Erne, I think also from the LOS project, she's also working with moisture and temperature. So she's actually trying to get the scale of moisture and the temperature effect. So from the LOS project, that will be one of the outputs.
Thanks again for broadening our perspective.
Thank you.